But it should not prevent. That should not prevent to pre-register the analysis anyway.
Okay, should we get started? Looks like this may be it. Anyway, so the goal for today's meeting was to learn what companies like Gold Standard Phantoms and Phantom Lab would are doing in terms of QA, and especially the cloud services they're offering to monitor system performance, and the phantoms they make that may be useful for reuse in the academic community. So big thanks to both Aaron and Richard for joining us and taking the time to give a talk and share their expertise and interests. Which one of you want to go first? Should I take the alphabetical order? Okay, let's go Aaron. So Aaron Oliver Taylor is an MRI physicist with over 13 years experience working in MRI. Began his MRI career as a PhD student under the supervision of Dr. David Thomas at UCL, building RF coils and various other bits of hardware performing ASL with separate labeling coils using parallel transmit, amongst having to figure out how to get a SMIS console to talk to a Siemens Trio, and was also introduced to the world of phantom building, both static and dynamic. And after his PhD finished in 2012, he continued at UCL as a part-time postdoc building coils and other bits of MRI compatible hardwares for preclinical studies, culminating in constructing 2 custom MRI large animal neonatal incubators that incorporated life support and physiological monitoring systems into a portable unit for transport between preclinical and clinical MRI systems. Wow, that's quite fascinating. At the same time, Aaron began working with Professor Xavier Golay at UCL on further developing the perfusion phantom from his PhD with the intention of commercializing it. A few prototypes along the way in 2015, they successfully obtained a grant which began Gold Standard Phantoms with the aim of developing a commercially available perfusion phantom for ASL. After successive grants and a round of private equity, the CASPER, standing for Quantitative ASL Perfusion Reference, was launched for sale in 2018 and has since been accompanied by complementary phantoms, which are sold to researchers and clinicians worldwide. As the Chief Technical Officer at Gold Standard Phantoms, Aaron wears many hats, sometimes juggling production and operational issues, providing technical support for customers, writing analyses and digital reference object software, managing scientific collaborations, writing abstracts, as well as leading R&D for new products. So I think this should be an exciting, interesting talk. So without further ado, Dr. Aaron Olivier Taylor. Are you speaking, Aaron? I can't hear you. Maybe you need to choose the right mic.
Yeah, I had it muted on the thing as well. Okay, right. Yeah, so thanks for the introduction. And just go back, start the presentation again. So, okay, so I'm obviously representing Gold Standard Phantom. So we're a UK-based company operating out of sort of London, although since obviously the pandemic, it's all been at home, but we have a small manufacturing unit sort of based in Kent. So as mentioned, we started officially in 2015 with an SBRI healthcare grant, which is a UK government R&D contract really to develop something. and they have two phases. So we got phase 1 in 2015, and then phase 2 in 2016. And this was for commercializing our perfusion phantom, which became Casper. So this down here is our kind of one of our last prototypes that we made to kind of test the proof of concept of it working. In 2017, we had an investment round. We had another grant, this time with the National Physical Laboratory and the National Engineering Laboratory in the UK. So these are the equivalent of NIST, really. And this was to do with the metrology and the modeling of our Casper Phantom. We finally launched the Casper Phantom for sale in 2018. And also we have another product called the Multisample 120. The fun star's here because I forgot to move it, but we did launch kind of an original version of the fun star, which was agar based in 2018, and then we had a few issues. So the official launch of the fun star was in 2019, along with our Spectroscopy Phantom and our cloud-based platform for doing phantom analyses, particularly in F-burn analysis, was launched in 2019. So that's just very imaginatively called GSP Cloud. In 2020, so we worked on a grant with the VUMC in Amsterdam and Mediri, which is a German CRO. And this is to do with the ASL and making it more standardized and kind of incorporating a combination of our phantoms and QA experience and the VUMC's ASL software, Explore ASL, and Mediri's expertise in clinical trials to try and create a bespoke product for clinical trials. And as part of this, we actually developed a digital reference object for ASL. So I'm going to quickly go over basically all of our products just to give you a general overview of what we have. So this.
Can I ask a question?
Yeah, sure.
Yeah. So you say digital reference object. Can you briefly explain or define it?
I've got a slide on it, but basically it's some software that will generate digital reference objects for arterial spin labeling. So it simulates the data and puts it in a format that is akin to what you'd kind of expect the data to look like when it comes from the MRI scanner. So you can use this to test your image analysis pipelines. And the idea is that it's very well tested and validated, so you can trust that what it produces is, you can trust it's doing what it's intending to do, and then you use that for verifying your own imaging analysis pipelines.
Perfect. Can you share it to the group?
The DR, yeah, of course. I've got a. Oh, you'll see. It's a few slides down the line, but we can. Yeah, I can show you more about it. So. Starting with the Casper Phantom, it's for obviously ASL for perfusion. It's got an MRI compatible pump. The idea is it delivers a perfusate, so a liquid, around the system at a known flow rate. It's got a calibrated flow meter in there, and we can get ASL images because we've optimized the time that it takes from where you label the bolus to actually reach the imaging volume. So that's on the same order of in terms of seconds as so between half second, 1.5 seconds is what you get in adults. And that's kind of important because you want to be able to do any kind of validation or sequence testing or QA with the same protocols that you use on your subjects or your patients. How familiar are people with ASL?
Well, I know the basics.
Basic. Oh, hi, Antoine.
Hello, Aaron. How are you?
I'm well, thank you. How are you?
I'm very good. Good to see you.
And you, yeah. Yeah. So so essentially, it's the idea is you have an area that you label, and you invert the blood or invert the perfusate that goes in, and then you wait some time for it to go somewhere, and then you image it, and it's kind of it's an endogenous tracer. So this has a few features that mean that it's very well suited for ASL. It can't be used for injection contrast agents. One of the questions we get a lot of the time is, can we put gadolinium in it? Because it's a circulating system, that's just not possible. So the main thing with this is it uses some porous material to simulate the capillary bed. And that's like our, I suppose, USP compared with, well, there's no other air cell perfusion phantoms out there, but that's how we generate the perfusion kind of signal, is by flowing perfusate through this. It's really like a sponge, but it's hard. It's sintered polythene, and that very nicely simulates the capillary bed, because the pores are sort of on the same sort of size as the capillaries, on about 10 microns. So yeah, a bit more about the Phantom. So this is just a cut through. We basically have an inlet, it then splits out, goes into these different layers of porous material where we have channels cut, and you end up with a signal that sort of blooms around each of these inlet arterioles. When we designed it, we used fluid dynamics, computational fluid dynamics, to optimize the design. So we got the right sort of flow rates in there, and then we built like a tracer model where we took the velocity field that you get from the CFD and then modeled particles, sort of like a modeling the tracer and seeing where it goes, and optimizing the time it takes to get into the imaging volume, that sort of thing. So we have something of a ground.
How stable is it, Aaron? Can I ask? Are we allowed to ask questions during the talk, or?
Yeah, sure, I don't mind. So the pump
how
itself.
How stable is it typically, and how stable is it regarding slight changes in positioning of the phantoms? Say, because in practical terms, conceivably, depending on the position of the phantom, these the velocities might change.
So the in terms of the pump, it has a servo control algorithm, a PID algorithm in there. So it will always deliver the flow rate that you set it to, unless there's some major problems going on. Like, for example, if you put the pump too close to the gradient, so the pump detaches a bit, it still sits on the patient couch, but if it's in the gradient field, it will vibrate because it's got an aluminium enclosure, and that will mess with the flow measurements, because we use a turbine flow meter, which is quite sensitive to vibrations. But so long as it's positioned far enough away, then that's okay. So in terms of actual positioning, the flow rates are such that the velocities are nicely within the expected range for getting good labeling efficiency. So that's not too sensitive for that. Although depending on the implementation of the ASL, some of them are sensitive due to your shim and all sorts of other factors that you can't really control for very easily. But most of the vendor product sequences now they switch their shims between labeling and acquisition. So that's not so much of a problem. In terms of orientation, yes, there will be some sensitivity to orientation. But a lot of the time it's a case of, with phantoms, I think, or positioning is quite important. A lot of people have custom holders made for them. And so when we did a round robin study, which I can show you here, where we took it around Belgium and the Netherlands, scanned at about 17 sites. And this is just the data from sort of 5 of the systems, which were all the same Philips system. They were all running the same software version, and the only real difference is that 2 of them were achievers, so they're smaller ball magnet, and 3 were ingenious. And we can see this this thing. Yes, I can. There we go. You have this difference in the mean. Basically, we just did very blunt kind of measures of the mean CBF that was measured inside these ROIs. And there is a difference depending on the system, but largely they are quite similar. And I think probably the majority of the difference is explained through things like labeling efficiency. But this was with me taking the Phantom to each of these sites, doing the setup, making sure that I was positioning it the same every time. And so my co-founder Xavier, we sort of tag team. He did it one week and I did another week. And I think there's definitely some differences in the ways that we were careful in how we set things up. But so the reason I like to show this example is because I did all of it, whereas some of them we had some splits between different sites, between him doing them and me doing them. And I think there's a difference, there's some bias. And I think also some of them we haven't got enough data points. This has 5, which is quite good compared with, say, just having 3 or 2 different sites, because there are so many different ASL implementations it's not possible just to directly compare. That was something we really wanted to be able to do. And at the outset of doing this study, we thought, okay, we're going to be able to get a really good feel for how ASL does between vendors, between different MRI scanners, across different sites. And actually really what it was with hindsight was it was a good pilot experience of seeing that there's a lot of fragmentation. And if you have everything that's the same, yes, you can make a good assessment between different MRI systems. But otherwise, it's actually quite difficult. Like this was also, we picked a protocol that was kind of easier for us to explain. So we did a 2D EPI acquisition rather than a 3D Grace, so there's less blurring, and that means that it's a bit easier to understand the data, because we have one factor is reduced, which is the point spread function that you have from the acquisition.
You said the main difference was due to tagging.
I think for this, the main is probably the labeling efficiency, to do with how well shimmed in the labeling region the magnets are. And that's something which, there's the larger bore magnets, the Ingenieurs are generally, the shim is not as good as the Achiever. There was one outlier, which was an Achiever in Utrecht, which had the old RF system. So, the 10-year-old more analog RF system rather than the D-Stream. And that had, highest signal to noise. And I think it used the 8-channel coil as well. We didn't even use the 32-channel coil. That had the highest, highest signal to noise, and, in some respects, the best looking images. I don't know if higher values necessarily mean better images, but certainly, and anecdotally, at the site, I was told that people favor that MRI system for various reasons. So, it picked up on that. Can't really necessarily explain it. I think that's part of the problem with using something like a Phantom, which is you're measuring the end result. You're measuring the ASL rather than looking at what's going on with the MRI scanner. It doesn't really tell. It's not very diagnostic of what those reasons are. It's just saying, are we getting these numbers? Are they matching? Can't even tell you about labeling efficiency. And one of the big problems is if you put the Phantom in, if you have a way of measuring labeling efficiency, then you can assess that. Say you could on Philips, it's quite easy because you've got a lot more control over how you can plan, and you can kind of piece together different bits of pulse sequences, so you can rotate the field of view and that kind of thing, and you could get a measure of the labeling efficiency. But that's only good for the Phantom. If you then go and scan patients, well, it's different, because every person that goes in there is going to affect the MRI system in a different way. So it doesn't necessarily help. But it's interesting as a base. I mean, ultimately, what you want to do is find out here how objectively how is the hardware doing if we try and do the same thing as close as possible every time in terms of what we're putting in there. So moving on to some of our other products. So we have, basically it's a standardized container, but we call it multi-sample 120 because it's 120 millimeters in diameter. And it's basically a sample holder that accepts 8 15 mil Falcon tubes. The samples go into the lid, they snap in, they snap out. You just, take the lid off and can easily swap them out if you want to. And it's kind of it's a lot of people tend to, if you're making your own Phantoms in a facility, a lot of people use Tupperware, or they use, try and cobble something together, and it's not very effective. And then when it comes to trying to do any sort of analysis, there's no model to register to or anything like that. So this was trying to have something that was a bit more standardized, and where you can use the the advantages of having a defined model. We've got some software which I'll show you, which we can generate nifty images from, say, 3D CAD files like STL files. So you can use that to register models too, which is quite neat if you want to, be able to analyze ROIs and things like that inside these regions. And
so, a quick question, Aaron. Is there like some kind of criteria that we can establish for what can be an acceptable hardware phantom? Because that's what you sound like you're implying.
I'd say there's some criteria if we were going to sell it. I mean, by all means, if you get something and you make it and it works for you, then that's acceptable. Ultimately, it's, you know, the proof's in the pudding. I think it's for me, I would say that there are some things like when you scan it, you want to unambiguously be able to identify the orientation it's in, where the things are, and not go, is this sample this value because I expect it to be that value, or is it, have I got it the other way around and something strange is going on? Because quite often if you just put a load of things in there and then it's got turned upside down, am I looking at a DICOM or a Nifty? Because those are showing things in, you know, the opposite direction sometimes. And it's not clear whether or not you're getting what you expect. I think that's quite important. I think that stability is obviously very important, but I would say it's more important to characterize stability and know when your things have changed rather than expect them to be to have longevity forever, because these things are made of plastic generally, and plastics are permeable to water vapor, and they're permeable to other kinds of organic stuff. They don't last forever. It's you know, I know we have phantoms that people keep for 20 years, like Branos and things like that, but they will change over time. If you can characterize that change and it's still acceptable, then by all means keep using it. If it's not acceptable, then it's going to be something that needs to be replaced. And I think if you look at any other kind of test objects or test equipment, that's standard. I think, you know, it's a bit of a funny market is phantoms, because you've got commercial offerings, and they're probably quite expensive. And then you've got people that build their own phantoms in research, and it might be you only need to build something for a one-off, or you build something which actually gets used for years. But they are comparable and they aren't comparable compared to the commercial offerings in different ways. And I think that's something to just keep in mind. Those are just my thoughts about it, though.
Yeah, so from where I stand, it sounds like we need criteria and standards, both to say when a phantom is acceptable for a typical goal and when we can compare across different phantoms, you know? Mm-hmm. So the digital reference object you mentioned before, that might come in handy in a way.
Yeah, I think
if I understand it correctly, you know?
Yeah, I mean, so one of the things I'm part of is in the OSIPI, which is the Open Science Initiative in Perfusion Imaging. And we have a task force which is about basically collating an inventory of phantoms and digital reference objects for perfusion imaging. And we're putting together some sets of sharing guidelines for if you want to share DRO data, if you want to share software, and also it's for sharing plans for physical phantoms. Because I think there isn't much in the way of a guideline, and like hardware is tricky. You know, there are some very good initiatives by like CERN for open source hardware, but that tends to be more about electronics, which are, I think, they're a bit more standardized in the sense of what exactly it is you're doing. Because it's, you know, it involves a PCB, it involves some components, you put it together in this way, there might be some firmware you've got to load on. Whereas for something which is I mean, essentially you need to give instructions so that somebody can recreate what's being done. And actually that has a lot of different aspects which are very variable. Like, are people going to be able to get the same materials? Like just saying 3D print this, actually, how do you 3D print something if the geometry is quite complex, so you need a special filament or that kind of thing? I mean, I'm not sure if 3D printing is the best process for phantoms anyway, just because the materials that are available are generally not very stable, and that needs to be managed. And I mean, for sure, if you have something which goes inside, say, a vessel which is made of a material that is more robust, and we know about the parameters of how long that will last for and that kind of thing, then that would be perfectly reasonable. But I think printing something and expecting it to be watertight, that's not going to happen. And also expecting, you know, if it needs some treatment, saying what that is. And I think it's just that needs to be specified in a way that isn't really reflected very well in papers. People put things on GitHub, that kind of thing. GitHub's more for software than providing plans for how to build something, but it is, you know, it's good for text-based things and sharing photos. You can put websites on there, so there's no reason not to use it. But I think there needs to be a good set of guidelines for how you can, you know, it comes in with reproducible research. How do you make sure that someone else can build what you've built and it's useful? I think that's really important, because you know, the commercial companies, yeah, will make some phantoms if they're shown to be useful and there's a market for them. But if it's just one or two, then quite often there's not going to be a market. It's not worth our time, and people can't afford what it would cost to do these one-offs, and unless people apply for a big grant, that kind of thing. But that's a totally different matter to trying to bootstrap something which everyone's using, you know, which is nice and inexpensive, and you want to share it. I think there's a very different, you know, that's a different model, but there needs to be the nice way of passing the knowledge on. I think so. I think that's important as well. And then there are a few other things which I think are like good practices for safety, like using safe materials, safe, you know, preservatives, not using sodium azide. That's one thing I'm really quite keen on, is people move away from using that because it's really horrid stuff. There are very, very safe and effective preservatives that, like, that's what we use, that are far better, and also will mean that you can easily ship them. So if you have collaborators, you can send phantoms to other people more easily. I think maybe that, you know, having advice on how to write a material safety data sheet for if you have to send a phantom to somebody, or send one around for doing like some sort of round robin. That's probably another good point for researchers. I think that, you know, those are the kinds of things. The actual defining what should a phantom do to be effective, it really comes down to what the purpose of it is. But there are loads of like things that sit around it which are to do with they're sort of non-functional, but they're important, and I think those things can be very, very nicely defined.
Yeah, I mean, from what I understand, I think there needs to be clear guidelines. For example, how can we compare the data from your phantoms to, for example, those from Phantom Lab, you know?
I don't think we can.
Yeah, is it? Yeah, if it's possible to consolidate them, that'll be good in a way, you know? So that's what I was thinking. So one thing I can definitely see is, you know, if it is possible to develop some sort of approval process for this, makes a reasonably good enough phantom to do FBurn QA, for example, right? Especially if you want to compare across different sites.
Yeah, well, I think I've got a bit to talk about kind of FBurn. So I mean, that's something to discuss, I think. But I think you know, there are different phantoms and they all do different things. Like you wouldn't do an FBurn with a water bottle phantom, for example. Really, I mean, people do, and then they come.
Yeah, I mean, I was referring to the most common case, you know.
Yeah,
there is definitely exceptions, of course.
Mm-hmm. Yeah, I think well, I think that if you can state a specific need, then yes, there could be good standards for that. And if you look at what's coming out of Kiba from all the quantitative stuff, then yeah, there are requirements, and they will set what the requirements are for a phantom. And generally, there'll be a commercial offering for that, but you don't have to. They should state how to also prepare a phantom that would be equivalent, and probably also verify that it is suitable. So I agree that's important. The main issue is going to comes down to certifying the materials, which tends to mean getting NIST to characterize them, and that's expensive, and then it becomes the same sort of cost as buying the commercial offering. So that's another issue, really. Okay, so just here's just some images of using the multi-sample when we were developing our fun stuff Phantom, just to kind of give an example of how we've used it. We have a new product, which is this enhanced multi-sample, which is the same vessel, but takes 24 vials. We also have add-ons for putting in fiber optic probes and things like that. This is a nifty that's created from the CAD model. So we have a little tool which I would quite like to release as open source, but it's sort of wrapped up in a load of other Python code for which, so it's not very easy to access at the moment. But it'd be nice to release this because I think it's quite useful. If you have an STL file, you can just basically throw this at it, or throw that at this for software, and it will generate the STL, generate the nifty. Although, yeah, it's so basically with this, you can in principle do a registration to your MR data, and you can have labeled regions and easily extract ROIs. So it just means that you can automate your Phantom processing. So if you've got some QA, QC kind of software, rather than having to manually draw ROIs and that kind of thing, or find some other way of segmenting or thresholding, that sort of thing, it should work quite nicely. This is just in the enhanced multi-sample. So this is when I spoke about wanting to unambiguously identify exactly where the vials are. So this is something we have in our enhanced multi-sample, which is having this kind of key that's engraved into one of the plates. We have two rods that extend throughout for where all the samples are. And so that means that we have effectively order one of symmetry. And we've also got a kind of like a little graphic which shows, depending on whether you're looking from the front side or the rear side, just where all the vials are in relation to the key. And I think that means that, both in terms of a machine identifying it or just a human looking at it, it is identifiable and it's unambiguous. And that's important to me because it stops me scratching my head when I have to look at the images and try and figure out which file is which. So we also have a spectroscopy phantom, which is essentially the Braino phantom with GABA added, and also not using sodium azide. And also you don't need to buy a GE scanner and their spectroscopy sequence to get one. So that was the other main sort of selling factor. It's the same kind of concentrations. GABA is at 2 millimolars. We have done them with other metabolites. So we've done one with 2-HG, for example, which was very expensive, but that was needed for somebody. And although there are certain metabolites which are not stable in water, such as glutathione, which we quite often get asked about, which is unfortunate, because I think it lasts for about 9 hours at room temperature in water, which is not terribly useful in terms of having a standardized phantom. So this is our equivalent of an F-burn phantom. So as I mentioned, we started out back in 2018, we got a request for 10 F-burn type phantoms. So we thought, okay, how hard can it be to make agar? And we ended up with some phantoms that most of them developed some issues with over time. We started with a slightly different shell. We made a polycarbonate shell. And as I mentioned, plastics, they all are permeable to water vapor by differing amounts. Now, polythene, polypropylene are far less permeable than, say, polycarbonate or acrylic. And what happened over time was that the water vapor would evaporate through the polycarbonate, and then the agar would shrink a little bit, stop being stuck to the sides, and then we'd get fractures in them, and there would just be cracks that would go through the phantoms. The other thing was actually to get them bubble free required quite a lengthy process. A lot of energy went into it in terms of just heating up. So, just to basically cook 5 liters of agar at a time was quite a lot. And, generally they're not terribly reproducible, particularly once you include the bubbles. So we started looking for a new type of gel that would be more reproducible and bubble free, and where we felt this was something we could improve on. And so what we arrived at was using polyacrylamide. And this is quite a nice gel because you basically polymerize the monomers. So you take acrylamide, you take a cross-linker, which is bisacrylamide, mix them together in water, add the initiators for the polymerization, and they produce a gel which is cross-linked. You can adjust the cross-linker, you can adjust the gel strength, and you can create gels which have different properties in terms of their mechanical properties. So we tried a few things out, and we arrived at something which we think does very nicely for an F-burn test. We use, as I said, we don't use sodium azide. We use a different preservative, which is this chloromethyl isothiazolinone and methyl isothiazolinone based. You can buy it from Sigma. It's called Proclin 150. There are a few different ones in the range, but this one is water based, and it's nice for phantoms, and you can use it about 0.1%. The actual amount of the preservative, so in the phantom, it's in the preservative itself, the actual active ingredients is only about 1.5%. So you end up at kind of micromolar, concentrations of the actual preservatives. So they're not detectable, because one of the main justifications that there are for using sodium azide is that there are no protons in there, because it's just obviously sodium and nitrogen. But this one is equivalent, and we've tested that. You're looking at NMR and not seen anything, even at 9.4 T. So just kind of to look at in terms of reproducibility of the phantoms, like this is just a typical one of our fun stars. We get quite nice, homogeneous, no bubbles throughout the middle. Whereas with an agar F-burn, and this is actually from the WTCN, from one that they've got, and you can see that there are bubbles, and that's because during the gelling or during the cooling, there's going to be some shrinkage of the polythene ball, and also of the gel itself is going to, the liquid is going to contract as it gels and cools down, and that just depressurizes, causes any residual dissolved gases, so particularly air, to come out of solution. Obviously, you can go to great lengths to try and avoid this, but it's sort of I don't know. Whether it's worth it or not, I don't know. A lot of people say that these bubbles are not an issue, and it doesn't stop you from being able to do an F-burn. I mean, that's been the standard. But if you want to be able to compare, the actual F-burn parameters that are obtained, the metrics between sites where you have different phantoms with different bubble patterns, is that possible? And the question to you guys is how competitive are you on your F-burn parameters? Do you kind of say who's got the best scanner? I was talking to Xavier about this, and he said that back 25 years ago when he was 20 years ago when he was in Baltimore at Peter Van Zyle's lab, they had one of the first 3Ts set up, and Peter Van Zyle was very competitive on maximizing all of the parameters in the from their F-burn analysis. Okay, so we have our cloud-based service, GSP Cloud, which does do an F-burn analysis. You essentially just upload the DICOM data from the study, select the image series that you want to analyze. So that it will identify any that are basically EPI time series, and then you just say, yeah, analyze these ones, and then it will generate a report for each of them, and you can plot longitudinally. So this is just kind of an example, and it produces a PDF report. So if you want to try this out, I'm very happy to make an account, and you can upload data to it and give it a go. And this is for anybody regardless of whether you have our Phantom or built your own, or you bought one from elsewhere, or you don't have any Phantom, you just got a bottle of Evian. So this is a bit more about our digital reference object. So this is essentially software that we've written in Python, and it simulates the I suppose the signal equations for arterial spin labeling, and then puts this signal into some synthetic MR images, and then it outputs these in BIDS format. So it's not DICOM, but it's the it's a consistent common format that then can be loaded into analysis software. And generally, certainly within research, most people are converting their DICOMs to NIfTY anyway. So this seemed a good starting point. To make DICOMs would be quite tricky. Need someone that really knows the DICOM standard for it to be able to cover all of that. So this DRO can be used as a general purpose MRI DRO generator. The software can, it doesn't have to just do ASL. So that might be of interest to you guys. So just to kind of give an overview of how it works, there's ground truths that are comprised of some images, some more parameter maps, and some configuration parameters. These all go into kind of the DRO kernel, which just generates the ASL signal, or that can be left out. And you generate some MR signal. At the moment, we just have basic spin echo, gradient echo, but this could be extended to have more complex signal models. There's translation, rotation, scaling to kind of simulate patient motion. We add noise in the Fourier domain to both the complex real and imaginary channels. So you get kind of a Ricean noise distribution when you have a magnitude image. We make a 4D time series because that's generally what we do in ASL, which comprises of an acquisition loop of control and label kind of a time series. And there's also this multi-phase loop. So in ASL, people generally do a single post-labeling delay, but there are also reasons why you would want to have multiple post-labeling delays, and that's what this multi-phase loop does. And that means you can then fit to the more full general kinetic model rather than just using a simplified quantification equation. And so just this kind of an overall of ASL DRO. So it's Python 3.7. The main libraries are NumPy, NeLearn, NeBabel. It's open source, MIT license. It's implemented with continuous integration. All of the functions and the underlying code is well tested and validated to make sure that it runs smoothly. We're very keen on having people collaborate. It's on GitHub. It's on the Python package index, and we've got a read the docs. At the moment, the most recent release is version 2.2, and there is an impending version 2.3 coming out. That's been coming out for ages, but I just haven't got around to it. But I would strongly recommend going on the develop branch and using that if you're comfortable with it, because there's a lot of new things that have been implemented since 2.2. And if you're on the documentation, then this would be to look at the latest rather than stable release, and you'll see the difference. So then I just want to touch in the final bit on our new sort of project that we started working on over the past few months, which is something which I think we're quite keen to get more involvement. And essentially, if we look at any kind of advanced neuroimaging technique, fMRI diffusion, ASL, spectroscopy, they're more sensitive to hardware changes. But the thing is if you are just looking at the images that you acquire, the raw data, you might not necessarily see any problems. And then you have the issue that the data might not be processed for weeks or months or even years until a study ends. So if there are any issues that would have been flagged by some sort of QA that was ongoing, but it wasn't, because the QA wasn't done or wasn't sensitive enough, then you might end up with large chunks of data that has to be discarded. It's waste time and it wastes money. So this is our starting point for trying to have something which is useful and our problem to solve. The other confounders really are that if we want to have site QA, then you need to have someone that is going to be dedicated to do that, and you need to be buy-in from all the people that are involved at the site. Typically to say that we can justify spending this time on a regular basis to do the QA. And I think that, as we're seeing, as a phantom manufacturer, is that people will end up getting to the point where they're kind of fed up of having phantoms. They're only going to be able to scan so many. Someone might buy one for a particular project, but in terms of what is being done on a routine basis, it's going to be the simple stuff that does more, rather than having a different phantom for everything, and then you end up having to spend 4 or 5 hours per week doing some sort of QA, and it's simply not worthwhile. And I think a really good example is the FBurn test for this, because it is standardized. The phantom is defined, or it was defined until we came out with a new phantom and said this is also appropriate. But it's at least it's standardized in the sense of what the phantom needs to achieve. The protocol is generally standardized, although there might be, over the years, people have deviated. There's an analysis which is standardized, and also generally people know how they should interpret it, at least in terms of being able to say that there's an issue with their system or everything's okay with their system. I think that there's probably a push now to try and have some more meaningful insight from what these values actually mean and what that might mean for the data you're acquiring. But that's probably slightly going beyond the scope of just QA. But I think it is important. But I think this is something that's also an interesting area to look at.
How much more do you have, Aaron?
This is just the last slide.
Okay, sorry to hurry you up.
That's all right. Or maybe no, I've got 2 more slides. Sorry. Yeah. So basically, what we're thinking is we start with our Funstar Phantom, because it's got quite nice properties, obviously for FBurn. It's got some magnetization, transfer, contrast. It has slightly restricted diffusion, so far less restricted than an Agar Phantom, but there's obviously no convection because it's a gel. So that makes it nice for looking at diffusion sequences. It's homogeneous because we don't have the bubbles in it, and we have better reproducibility between phantoms. So you've got better comparability between sites with different phantoms. And obviously it's a good size for coils, for head coils. What we want to develop is an objective standardized measure of the MRI scanner's performance. We want to have probably vendor-specific procedures and scan protocols, because there will be differences. We want to standardize our analyses, make sure that they're well tested, well validated, well documented, and make them available. And we also want to eventually get to a point where we can specify sort of benchmark thresholds for what's acceptable for neuroimaging data quality. So this is essentially our goal. We're going to develop protocols, make these freely available. We're going to try and recruit for trials at multiple sites for people that want to try and do this kind of scanning on a weekly basis and contribute. And then eventually we'll get to the point where we have a version 1 release, the publication, which we can keep live. We'll keep on updating it, and then if there is a want for this is something we can include on GSP Cloud, or we can include on, so there was this, there's another service which actually came across through you guys. Is it Brainweb, which is another, these are other places that can have this kind of thing included on. So that's where we thought that would be a nice to get feedback on and see what whether there's any interest from you guys. So I will stop there.
Any questions, comments?
Hi, Aaron. Thank you for the presentation. Pradeep invited me. I was curious, have you done with the MRS any QA? I mean, like what have you used? I mean, like I missed the beginning, like probably the first time minutes of your presentation, which I think was great. But with the, so it was mostly ASL, I think I get most of the work you've done. But you also mentioned some ASL, some MRS phantoms, the pretty much the Braino with the improvement GABA and H2G added, right?
Well, the 2HG was a one-off.
Oh, okay.
Yeah, it's too expensive for us to be in a standard phantom.
But with the Braino Phantom, then are you doing currently any work like any QA or?
Well, I mean, we're a phantom manufacturer. We don't have
MR
systems ourselves, but we have lots of customers that are using them.
Okay, yeah, because G is probably not selling. I mean, I think we have here a couple of Braino 20-year-old Braino Phantoms and stuff. But you're providing if somebody needs an MRS Phantom, they would come, would reach out to you for a similar Phantom. What do you call it? I think it's
Spectroscopy reference.
Oh, okay. And the difference between the one you're making and the old G Phantom is yours has the GABA added.
It has GABA and it doesn't use sodium azide.
Okay, sounds great. How much the software usually?
The software?
No.
Oh, they sell for. Sorry.
Yeah, this Spectre Phantom.
So the list price at the moment is £2,000.
Okay. You guys give discounts? I mean, if you like the site better.
It depends on what exactly the the requirements on the needs are and that kind of thing. Yeah, we have sort of a discount policy, but yeah, we're pretty kind of open and standard with our pricing.
Okay, sounds great. Yeah, no, I mean, like because I'm mostly interested in fast MRSI, so I kind of, it wasn't my main interest to develop a QA procedure for using MRSI, but a group asked me if I can do that, so I took my fast MRSI acquisition, because I do agree, I mean, like it would be more sensitive to to scanner hardware malfunctions and so on. So I actually presented an abstract at the ISMRM this year using, and I have the software, I mean, like the programs to reconstruct the data automatically. You need the C2P to install it on a Siemens scanner, the sequence to run, but then the reconstruction would be automatically done, and that's all that's needed. Spectroscopy Phantom, like Braino. So that's where my interest in the phantoms and came from. But yeah, thank you for the talk. I mean, it was very interesting.
No problem. Thank you.
So if there are no more questions, I have a ton of interest in continuing the discussion towards actually developing some sort of guidelines / standards. I think there is a lot of interesting scope. Now, so the Neuro QA, is that some kind of group you mentioned, Aaron?
I mean, we're calling it that, but basically it's a starting point where we've done some requirements gathering. We've spoken to various people about what we think would be, what the problem is and what we are trying to, what we feel we try to address. I mean, from to be completely open, for us, this is a way of adding more value to our FunStar Phantom.
Yeah.
Because obviously, as an F-burn Phantom, I mean, the Easy FMRI sells them, or people can make them themselves from AGAR, that kind of thing.
Yeah.
You have to have a good justification for wanting to have, a R1, which would be, well, there's added value. You can use it for these things as well. So that's our reasoning, but also just it's useful. It helps. It brings QA to the forefront, and it's important.
Yeah, I mean, from my perspective, that sounds like the special interest group we are part of. I would request all of you to join and share and whatnot. There's other people who are interested to contribute to. So I'm mindful of the time. Go ahead, Cyril.
Just quickly, from an acquisition perspective, when you look at what, for instance, Aaron was showing with the F-burn, It's like I often notice that on QA, people tend to do easy sequence. Whereas in participants, we don't do that. We use all the gizmos. We just do, fMRI with an SMS at 6, 8 TR of half a second. We push everything we can on the machine. And then behind this, somebody comes like, I'm going to do QA on my board with a TR of 2 seconds. You're like, nobody does that anymore.
Mm-hmm.
You want to use the same parameters as what you do in your application. Seems to me, and I see very often that, the QA is, even like on the T1, they would do a good old-fashioned T1, whereas this is, not necessarily what you would do. You would push your MPR edge, your MP2 edge at max, or, it's funny that the QA typically doesn't follow the application. It seems to me very weird, because obviously you're interested in exactly what you do on people, not on, I don't know. It's just like a remark on, when I was looking at the F1 protocol, people are we actually still scanning at TR 2 seconds?
I suppose the question is, do you need to scan with the same sequence as on your subjects to be able to pick up, the kinds of instabilities and things like that? Is it diagnostic as it currently is,
for instance, right? You typically have in plane and in volume acceleration. Right? You would have your grappa on Borosiemens, and you would have an SMS to get multi-band acceleration. But you can have, if your gradients are not doing a good job, the pulses are not sent properly, you would have crosstalk between the different pulses, right? Between the different slices, especially when you accelerate, when you do like, SMS 8 or obviously SMS 2 is fine. So you could pick that up in the Phantom that you couldn't if you do it with your standard, no acceleration, TR 2 seconds.
But might that muddy the other things in an FBAN? Might it be that you need a separate test for testing? I mean, these are the things to explore, I think. I think you're right. But the question is it comes down, it's like the ASL Phantom, we
yes.
measure ASL everywhere and we can see there's a difference, but it doesn't tell you what the problem is. And it could be that it muddies it, or it might not, it might be fine, I don't know.
Most people use Pascal ASL, and then they would not use that sequence on the Phantom. You're like, yes, but that's the way you do the tagging on, in patients.
Well, we designed the Phantom so that you would do the same thing as you do on your patients for that reason.
But you got my point.
Yeah.
All right, if there are no further questions, let's move to the second exciting talk from Dr. Richard Mallozzi. I hope I'm not murdering the name.
No, that's good.
Yeah, he's a physicist at the Phantom Laboratory Incorporated, where he's been developing phantoms and software for MRI quality control since 2014. Richard received his PhD in physics from University of California at Berkeley. Prior to joining the Phantom Lab, he worked in the field of MRI for over 15 years at GE Global Research, GE Healthcare, and ONI Medical Systems, where he worked on diverse applications, including MRI system development, MR quality control, neonatal MRI, and interventional MRI. So I'm quite looking forward to his talk and the services offered by his company. So without further ado, Dr. Mallozzi.
Thank you. Let me get in presentation mode here. All right, can you all see my, do you see my slides?
Yes.
Okay, great. Thank you for that introduction, and thank you, Aaron. That was a very nice talk you gave. So I'll tell you a little bit about the Phantom Lab. I know that we're a little constrained on time, so I'll try to be brief with much of this. Phantom Lab was founded in 1989, has 22 employees, and it's located in a rural setting in upstate New York. It's built the building is a very energy efficient building. I don't know where all these lines are coming from here. It's not a net zero building, but it was built with a lot of the principles that were put into a book, a well-known book by William McClay.
Hold on, are we being Zoom bombed here?
Yeah, I'm seeing all kinds of strange lines appearing on my screen.
Hey, who is Tim Wilson? Should I ban him?
I'm erasing the stuff that was just drawn on there. You can use an eraser feature.
Huh, okay.
Oh, sorry. Who is Tim Wilson? Sorry. Okay. Because I wasn't sure if he's one of the researchers. Anyway, sorry. Go ahead.
Okay, I'm going to go on to the next slide. So the Phantom Lab makes a number of different products for different imaging modalities. The majority of the business is actually in the CT Phantoms. We make the industry standard CAT fan series of Phantoms. On the MR side, we make a range of mostly systems-oriented Phantoms, and with a particular emphasis on geometric distortion. So we sell a lot of Phantoms into the community of radiation therapy planning. These folks are particularly interested in distortion and are grappling with the interesting question of how to use MR, outside of the traditional radiology environment. So briefly, what's in the talk, I'm going to talk about the Phantom Lab Phantoms and software, some of the challenges we see in MRQC sort of at the street level, and some experiences.
Richard probably has a huge cock.
I guess we are being Zoom bombed.
Yeah. I apologize. I removed both of them.
Mm-hmm.
Maybe I'll put a password. Go ahead.
So, and then I'll share some experiences we've had working with radiation therapy planning departments and a little bit of former ADNI participants. All right, some general challenges in quantitative MRI. This is a standard set of things you all are more than a little familiar with: variation in protocols and scanners, underdeveloped QC methods, lack of historical importance of quality control in diagnostic radiology. These are standard items. I won't dwell on these. I might just point out the last one, that there's a lack of a strong QC culture within diagnostic radiology because of the historical dominance of non-quantitative applications. That's actually quite a barrier to introducing these techniques more widely. So on the practical street level challenges, one simple one is that quality control is expensive, time-consuming, and boring. Right? That's one of the reasons that people aren't doing more of it. Another one is simply, what do you do with the data? A lot of departments, especially in radiation therapy planning, know that they need to do more QC than what's done in diagnostic radiology, but it's not really that clear to anybody what exactly to do with the data. MR and this comes from the fact that MR systems are really quite complicated systems. There's quite a variation in the sophistication and the interest in the users. If you're trying to do what we're trying to do, which is sort of bring routine QC to the masses, well, those masses vary quite a bit in their sophistication and interest. If you're making a software product, for instance, for people who are primarily scientists, they're going to use that software in a way that's less error prone, and it can be a little bit easier to handle the range of use cases. Another challenge in this is the ongoing support that's required. MR data is quite diverse, which means if you're trying to run QC on a wide range of MR data, there's a lot of different ways that the data can come in and sort of throw an analysis off, and that has to be, somebody has to go through that and diagnose and figure out what is going on, and there's quite a lot of effort that goes into this. So ways to address some of these challenges, we put a very heavy emphasis on workflow. The tools have to be extremely easy to use. And for us, this often means we're trying to do a lot of things with just a single Phantom scan and a fully automated analysis. There are a lot of things you'd like to measure that can't really be done well in a single scan. But you really quickly run into a barrier of, if you make the workflow complicated, it's not really going to be used on a regular basis. So, to wherever possible, we have limited ourselves to single Phantom scans, although we are working towards enabling multi-scan tests as well for those who want to do that. It's also important to recognize that for this to be used routinely, you have to offload as much of the work as you can to a technologist rather than a physicist. The physicist, ideally, you would want them to review it, if something has gone wrong, or if they want to do it more often, fine. But you want the scanning and the uploading, as much of that as possible, to be done by somebody who's not a physicist, because it's simply too expensive to have physicists do this all the time. To that end, we try to develop our software with layers of sophistication, starting with just simple pass fail. First question is something wrong with the scanner or not? That's the first level question. If something is, we have increasing levels of detail for specialists to look in and figure out what's going on. We think it's important to view Phantoms and software as an integrated product. As Phantoms, as we try to make Phantoms that tell you more and more about the scanner, there's a very heavy analysis component. But also it's important to design, Aaron touched on this a little bit as well, it's important to design the Phantoms in a way that enable the software to analyze them automatically. This means putting in features so that you can get the orientation and enable the automated analysis to be performed. And then a way simply for the community to share best practices. And I think that's something that is particular interest to this group. All right. So a brief overview of our Phantoms. Historically, a lot of this work started with the original ADNI Phantom. I actually developed this Phantom when I was working at GE Global Research, then called Corporate Research and Development, way back in 2001 and 2002. And worked with the Mayo Clinic to get it into the Alzheimer's disease neuroimaging initiative. It's mostly a distortion Phantom with a little bit of signal to noise. The ADNI/Mayo Clinic provides an open source code for analyzing the Phantom. Our company, along with an associated company, ImageAL, have a commercial analysis service for it. And we've continued to support some people who either want to do something similar to the ADNI analysis, or they participated in the original study and want to keep some of that going, but don't want to run the analysis themselves. So I'm mostly going to talk about two of our newer Phantoms. One is the S for System 162, that's intended for head coils, and both of these are systems Phantoms with heavy emphasis on distortion. The other is intended more for torso imaging. This is the MagFan RT. This is a large Phantom, and one of the major challenges in trying to measure distortion on large volumes is the Phantom can get extremely heavy if you're trying to cover large fields of view. And there are different ways of approaching this problem. Our solution has been to divide the Phantom into multiple components. But that introduces a very significant challenge, which is that you can never rely on the location of each component to come together perfectly. So you have to be able to distinguish between residual rigid body offsets of one module relative to another from an actual geometric distortion. And this is a problem that we've solved, and I consider that sort of the sauce that makes this technique work. The Phantoms are generally filled with plastic with water, just a background fluid of water, apart from some contrast spheres. They're fluid filled with just a single type of background fluid, and most features appear dark because they're solid plastic. So our general aim is we're trying to provide a very thorough systems check with a single Phantom, just to keep things simple. We want to implement sophisticated quantitative measurements wherever possible. We want it to be trivially easy to use with a fully automated analysis, and provide longitudinal tracking automatically as the data comes in. And also, we think it's important, and this gets to some of the discussion we were having at the end of Aaron's talk, that we think that it's important to have an aspect of QC that runs on the clinical pulse sequences being used, not only on a fixed set of sequences. I'll touch on that later. So these are the measurements that we perform with the Phantom. I'm showing one of the two, but they both perform a similar set. We measure nonlinear distortion, slice thickness, resolution. With regard to those, we have multiple features in, so that if the slice plane falls on any of the cardinal axes, we can analyze it. This Phantom actually only covers two of them. The body Phantom covers any three of the cardinal axes: axial, coronal, or sagittal. Three-dimensional uniformity, signal to noise. We have recently introduced a set of contrast spheres to get an array, just 12 spheres that give you a range of T1, T2, and ADC values. And then laser alignment to compare the laser alignment. This is more aimed at radiation therapy. A laser alignment with the location, the alignment of the therapy system. So before I go deeper into the analysis, I want to say some general words about MR quality control. We see that there are two facets of MR quality control, and this goes to the discussion that we're having at the end of Aaron's talk. The first is the question of is your scanner operating correctly, or is there some problem with the scanner? And so you're probing the hardware, firmware, the scanner software. And for this, you don't need to use clinical conditions. You can use a set of standard sequences to probe the system to see if something's wrong. There's another aspect which is more sequence specific, which really gets into how the scanner is used and also answers questions about a specific sequence. Sometimes you want measurements on the specific sequence you're using. In our case, it comes up, for instance, with distortion. How large is the total distortion on the particular sequence that somebody wants to use, because they're going to draw contours on a tumor and they need to plan their margins. So you may have specific questions. And also you want to catch possible user errors. Maybe somebody came in and changed a parameter. For instance, you may have a sequence that's been qualified as nice low distortion, and somebody realizes, oh, you can lower the bandwidth to receive bandwidth to improve the signal to noise, not realizing that that has implications for distortion. And so, testing things like that can catch user errors or configuration errors. Perhaps the distortion correction along the slice direction isn't turned on. That's often that's an option, which can affect the distortion quite severely. You may have specific questions about resolution. So for studying these kind of questions, you really want to probe the clinical sequence as closely as possible, and we've aimed to do that with the way the features are designed and the background solution that we really try to do the very pulse sequences that are being used in the clinic. All right, so a little bit more about these measures. I'm going to go through some of these measurements and give some details because some of these we do in quite a different way. With distortion, just background, a common way that distortion is measured. This is the ACR Phantom, for instance. It's just a single measurement of a single distance. And there are a lot of shortcomings in this. It's just really measuring the scaling. It's not really measuring spatially dependent distortion. It's not really all that accurate. And it's often done manually. There are frequently automated solutions, but a lot of people simply do this manually. We do this quite differently. We put in hundreds of fiducial markers. They're typically 1 centimeter diameter spheres that can be segmented to usually around 15 or 20% of a voxel dimension. And we calculate a full nonlinear 3D distortion. We have quantitative outputs for pass-fail tracking of some of the distortion values, and then graphical outputs for more in-depth analysis. One thing can be done, for instance, if you see this red line in the lower corner, this is a 3x3 plot of the distortion. And distortion is something you can't fix that much in the clinic, but there's one adjustment that they can be in, which is gradient calibration. When the manufacturers calibrate gradients, it's typically to about 1% accuracy, which can leave you, easily with a millimeter distortion, over a 10 centimeter radius. That's for many applications, that's fine, but when you're planning cancer therapy, that's a pretty big deal. And so a scanner can be in spec, but still leave a significant fraction of a millimeter in distortion. And using this data, you can actually calibrate the gradients much more accurately than what the manufacturer can do with their techniques. And that is starting to be done a little bit and enables people, for instance, to reduce the margins on the radiation therapy treatment, which spares more of the healthy tissue and a cancer treatment. The software we present in layers. So for instance, I mentioned this before, the first is just a summary of did anything fail, pass, fail for each of the different types of tests. And that's often all somebody wants to know. For some people, that's all they want to do. Did anything fail? If it did, then, or also that we provide quantitative measures. So you can pick any of these numbers and have the software track them longitudinally and set up pass-fail criteria so that if they go out of spec, you get an email and get alerted. We provide some graphical output for looking at things in more depth. This is a plot of distortion versus radius of the fiducial from isocenter, a quick way to get a read on how large the distortion is within some radius. This 3x3 plot is similar to what was used in the ADNI. It's for understanding the spatial pattern of the distortion. Where is the distortion highest? And then for any of these quantities, you can click on it and automatically get a longitudinal plot of which you can set tolerances and get alerts set up. A somewhat related measurement is the laser alignment. So one of the things that the analysis does is determine very precisely where the phantom ended up. So if you carefully align the phantom with the laser system, then the deviation from that tells you how far off it is. This has turned out to be a very important test for the radiation therapy community, especially for these combined therapy and MR machines. For instance, one thing that was discovered with this phantom on some systems is that as the gantry holding the source moves around the MR system, it shifts something. Perhaps it's just a magnetic field disturbance, or maybe it's moving, moving some other part. But they noticed that the apparent center of the field of view was moving by close to 2 millimeters as the gantry went around. That's a huge number when you're planning cancer therapy. So that was discovered with this with this technique, and this is becoming one of the more important test elements among the customers who are doing radiation therapy planning. Resolution. A common way that resolution is measured, such as in the ACR Phantom, is with these series of dots that are interleaved. The idea is that the user looks at the pattern of dots and tries to figure out when they can resolve dots, and then sort of makes a judgment there and decides what the resolution is. There are a number of shortcomings to that. It's subjective. It's difficult to adapt to automated measurement, and it's rather limited in its range of measurable resolutions. So we've taken a very different approach. We're measuring the response functions, the point spread function, the edge spread function of the system using circular apertures. The idea is that we acquire an image of the aperture, and you can look at that as an extended edge, and from that we can determine what the point spread function is. And so we have 2 of these features in this Phantom, 3 of them in the other, so that you can pick it up in different planes. So the idea is that you start with an ideal circular aperture. We know what the dimensions of the original were. We start with a candidate point spread function. We blur the image mathematically to get a fitted filtered image, and then compare that to the actual MR image of it. And then we iterate, changing the PSF to get the best match between the filtered image and the actual MR image. And you can see on the left where we subtracted, this is actual data. On the left, I'm window leveling to the same level as the original, so it looks completely dark relative to the original scale. If I window level it up so you can see the noise, we don't quite get to the noise level, but we're down pretty close to the noise level on that subtraction. And so from that, we can calculate the point spread function or related quantities like the modulation transfer function or the edge spread function. So these are often useful tracking or qualification measures. Probably the most useful single measure, I would say, is the edge spread function, because most of the time structures that you're looking at in MR, the edge is more like an edge than a point that you're looking at. So this is probably the most real characterization, or most practically relevant calculation of resolution for an MR system. And this can have this can vary quite a bit. There's a trade-off, of course, between trying to make a sharp resolution and ringing that you get. So you can actually see that pretty well with these measurements. You can see on the left a higher res sequence that reconstruction, I should say, that introduces a lot of ringing. A lot of times a sequence will deliberately be smoothed in some way, but the nominal resolution will be reported is being quite a bit lower, and that can be off by a factor of 2. So for those drawing contours, that may be very relevant to know whether what is the real resolution of the system. To illustrate where this could come in to play, if you're drawing a contour around structures such as outlining a tumor or in volumetric MRI, where you draw that line, it's a very fair question to ask. Where would you put that line? If you're outlining a dark structure, and here I'm showing an interpolated version of a blurred edge. If you're outlining a dark structure, are you going to put that line closer to the dark or closer to the white? If you're outlining a white structure, where would the human eye tend to put that line? Well, if you're talking about a 2 millimeter edge spread width, just that kind of difference could become quite pertinent to what you're doing. And so knowing the width of this edge spread function, might be something that would be used for qualifying a pulse sequence for contouring. I'm going to move on to uniformity. Usually uniformity is done with just a single slice in a single plane. Or your alternative is to have a large dedicated phantom that's nothing but a big, empty ball of fluid. We have a different approach to this. We're able to measure uniformity in 3 dimensions with this phantom, and we do this by using what I call virtual spheres. So virtual spheres are not real spheres. They're just places we know where there are no plastic features. It's just background fluid. So we can sample the background fluid in hundreds of locations, and uniformity tends to be sufficiently slowly varying that you can characterize it pretty well by just sampling it in a large number of places. And so we can fit this over 3 dimensions and use this as a quality control metric. And in particular, what this is good for is trying to detect one of the most common failures in an MR system, which is a failure of a phased array element. We show 2 measurements. One is the normalized standard deviation. That's just the standard deviation of those measurements divided by the mean. And then we have something we call normalized spread, which is the difference between the 10% brightest pixels and 10% dimmest pixels divided by the mean. And that's more locally sensitive. So if you have a failure of an array element, you might pick that up more sensitively with this. Here's a clinical example where it detected a failure. One of the elements failed, and it was caught with this test. Now you can look at the image and say, oh, well, if you can easily, it's pretty obvious looking at that image that it's there. But the point is that these tests are not usually done by the physicists. In this case, it's done by people who don't know anything about MR. They're therapy planners, or it might be an MR technologist. And it's surprising how they look at an image like this and nothing will strike them as odd about it. So to have a test, to bring the physicists out of having to review it every day and detect these kind of problems is very valuable. Signal to noise is another common way of detecting problems in the receive array or in the transmit chain. We do a very simplistic signal to noise, again, trying to keep the workflow simple. This is just we have dark regions in there. We take the signal near the center of the phantom and divide it by the variation, the standard deviation in the dark regions, and we sample those in a few regions around the phantom. I certainly am aware this is not the best way to measure signal to noise, but from a quality control perspective, where you're trying to detect a problem with the scanner, it's very powerful for finding problems. We are moving towards enabling more sophisticated measurements, multi-scan types of measurements, but from a failure detection standpoint, this is very valuable. Again, as I mentioned, as these measurements come in, you automatically get longitudinal tracking off of which you can set tolerance parameters and get alerts. We've recently introduced a set of contrast spheres. This is still somewhat under development. We're working towards getting NIST traceability. We're not there yet, but these are made of a combination of copper sulfate, manganese chloride, and PVP to sample different ranges of T1, T2, and ADC. So I'm going to move on to some practical challenges in the field in general. What we see in this is that there's a significant disconnect between the appetite to spend money on QC and the cost of actually providing Phantom software and support. Phantom is expensive, the software can be expensive and require a lot of ongoing support, and more so than there is an appetite for spending money on that kind of thing. Software development and maintenance costs are high. It's not just the development, you have to have training and support staff in order to have a product out there. Sites often need a lot more than just the base analysis software. They need an integrated system capable of managing multiple scanners and often on different campuses. You have to have account management stuff, billing, all of these things. And also it's a challenge. MRI data is very diverse. If you're trying to make a product like we're doing where you can run QC on a wide variety of sequences and bring it to the masses, that diversity of the data is a challenge in itself. And then this is this is an unfortunate one, but there are a tremendous amount of user errors when you're trying to bring it to the masses, that all have to be diagnosed. Simple errors like uploading a scout scan instead of the actual scan, or uploading an image of nothing but noise. They didn't scan the entire Phantom, or they didn't upload the entire series. Some slices were missing. The wrong Phantom. We get a Phantom, that might not even be the right modality. Oops, I have that one in twice, speaking of user errors. And then a general question is, what do our people to do with the data? As we sell this product, we are actually not allowed to make recommendations on specific criteria, because to do that, we would have to have validated, here's a set of criteria that, that works. Well, the applications are so diverse, we can't possibly do that. With former AD&I participants, a lot of them get the Phantom and they use the analysis, and they're largely just picking a few parameters, taking a baseline, getting the variation, and picking a few parameters that they watch carefully. But most of them are not doing a whole lot with it. The radiation therapy community is kind of an interesting one because they have a strong history of QC. It's a very strong QC culture. They already have a department that's dedicated to running QC on all of this very important equipment that's going to be applying the radiation therapy. So as they start using MR, they already have this strong emphasis on QC. They're asking, how do we know that the MR scanner is working? What tests do we need to do? And the idea is that a tool like this is put out there. We're not telling them exactly what to do with it, how to do it, but as a community, they figure it out. Sites start working with it, discovering how it fits into their workflow, what problems they're able to detect in their equipment with it, and they publish the results, they share the results, and gradually gets adopted by more people. Eventually, a task group is formed and recommendations are made. But it's really the culture that I think that we'd be looking for in quantitative MRI to bring that QC culture. It's natural in the radiation therapy setting because the errors are so critical, whereas historically in diagnostic radiology, most things don't matter that much. Radiologists can read through all sorts of problems until quantitative MR comes along that changes that. It's just not big enough yet to have moved things on that. So in summary, our focus is really bringing routine QA to the masses. We have a heavy emphasis on workflow and ease of use. And I think that the QMRI community could look towards radiation therapy planning as an interesting precedent for how quality control is put in more widespread use. So thanks very much for listening.
Oh, that was fantastic, Richard. Thank you so much. Any questions?
Richard, I was thinking just what you finished by, which is the quantitative MRI. Obviously, R1, R2, is becoming more and more popular. So, what kind of things, what kind of QC and what kind of phantom we need for that?
You mean for running QC on T1, T2?
Well, if you want to compute R1, R2 maps, right? Mm-hmm. Which is basically, because it's a bit like what earlier Aaron was talking about ASL. The idea is that, this is quantitative MRI and this is reproducible between sites. An R1 map, the values should be the same no matter if I run it on a Siemens, on GE or Philips. And even though we know it's not exactly the case, but that's the idea, right, behind quantitative MRI. Yeah. Because now we are measuring relaxation time, right, in T1 or in T2. So this value should be, they should be the same.
Yeah.
But it means we need dynamic phantoms for this kind of stuff.
Yeah. I think that in each of these, we have to start with the end application and work backwards and see where is that going to lead, because that can lead to a whole range of different phantoms. To really do it properly, and looking at all the endpoints that you're starting from, that you're trying to figure out and go backwards, is there even one solution that that would cover a wide range? There may not be. So I think then the question has to be asked is, what is going to be practical? It's one thing to do it with a few people, a few scientists who are trying to do it for themselves and a few others. But if something is going to be brought out and be used very widely and done on a weekly or even more frequently basis, and for a wide range of things, it's a real problem having all of these different tools in there, all these different phantoms that need to be done. And so, we don't see ourselves as the right people to answer that question. We enjoy working with the community to work towards that, and our products kind of derive from that. Actually, these products really came to Phantom Lab before I was the seeds were sown before I was at Phantom Lab. I came to Phantom Lab while I was working at GE, looking for a Phantom to do certain things. And I think that's where we have to come at it, starting with the end application, working backwards and saying, can we define a set of specs, and how broad of a set of applications can we cover with as simple a set of tools as possible? Because the complexity and diversity of the tools is a major barrier to more widespread use of the QC, and there's going to have to be compromises between how broad a range of applications you can QC and the quality with which you can do that, for each one.
I mean, I think it will ultimately, as you were saying, be a lot of related to how it's used in the clinic, right? So if you think, for instance, now you can have I don't know on the other scanners, but on Siemens, right, you can have an MP2 range. So basically, quickly computing your R1, basically your T1 decay, and I ran some I ran some QA, and also like on myself with like spin echo 20 minutes for one slice by doing many measurements, and you do the same thing in 4 minutes with your MP2 range from Siemens, and they get like the exact same values. Right, and so we were like, okay, we know it works, but then because we didn't have a phantom, so I was the phantom.
Yeah.
But ultimately, because it's not used in clinic so much at the moment, but we can easily think that these are sequences, because they are very fast to acquire as well. This would not be a problem. I can see them being adopted.
Mhm.
But then because this is quantitative, clearly you need to be able to say, hey, this is computed properly, right? The scanner is measuring things as they should be.
Yeah. Yeah, and I think it's always going to come back to working backwards, starting with your end application and understanding the sensitivity to the parameters that you can measure with a phantom, and then setting specs on phantoms. So I don't. I think to standardize in some set, we have to come up with phantom specifications or solution specifications. We need this % accuracy with this kind of stability, this kind of temperature, either temperature dependence or a means of calibrating for temperature variations.
Actually, we've seen that on the T1 values, measuring on one of the phantom, because we had changes in temperature over several days inside the scanner. Then, of course, the speed at which it was decaying was slightly different, several milliseconds. But then we were like, oh, yeah, but crap, we didn't control for temperature. But we recorded them. So then we looked at it and we were like, okay, now we can, yeah, I mean, it's crazy.
Yeah, well, it's nice that the human body is a temperature-controlled source. So you can use yourself as a phantom.
Yeah, so this goes back to the small discussion we had in Aaron's talk too. I think it sounds to me like the phantom spec you just mentioned, Richard, is something to be looked into by the group. What is an acceptable phantom, say, to do fMRI studies?
Yeah.
And I guess the length of the study matters too. Over the year, right?
Yeah.
So the hardware certification that Aaron was mentioning, that comes into play too.
Another thing that is needed out there is some way of probing phantoms after they've been out in the field for many years. Because after a phantom leaves our hands, it's out there for many years. And unlike most systems, which have some way of checking on it, a service engineer or something, phantoms are out there. We don't know what's going on with them anymore unless we're doing the analysis. So some way of turning around. Sometimes you can have an MR protocol that, although it's not something you want to do frequently, it can be made to be much more accurate than a typical clinical sequence. And if you can turn it around and probe the phantom to know when your phantom is starting to have problems, just as Aaron referred to, eventually, solutions can leak into plastic, get mixed, you lose your contrast, something can go wrong. The phantom themselves need quality control over time, to know, because it's a very difficult problem to put a phantom out there and expect it to work for 10 years. We have phantoms from the AD&I that are almost 20 years old now. People are dusting them off and trying to use them again, and there's a lot of problems with these things, right? And yet it's too expensive for them to fix it. They don't have the budget for that. A new phantom, our modern phantoms, are too expensive for them to buy. So we need methods that once the phantoms have been out, you can probe the phantom and run a quality control with an MR scanner too.
Right, like I guess the phantom drift, in addition to the system drift, so to say.
Yeah.
That comes to the question of cost. You just talked to that. Do you guys have numbers? Would you probably send us quotes to the group or here?
Yeah, I mean,
because just a second, Cyril is trying to. You want to talk about the data collection effort, Cyril and Derek? Cyril?
Yeah, I mean, it's just because obviously we're interested. Well, T1, T2 is the standard stuff, but we do mostly here functional MRI and diffusion. And so, this is the stability of the scanner over time, for instance, is a big thing. Obviously, for diffusion, we also have like distortions. Phantoms don't move, and so that's something that's different from humans. But like homogeneity over space and things like this are also interesting. And Yarek's been doing some work recently where I show how the phantom can be used to correct some aspects of what we can see over subjects over time. So, variations somewhere in the scanner that are captured by the phantom can be regressed out on subjects to remove some noise. We don't know exactly what it is, but clearly, if it's on the phantom, we know it's something due to the scanner. And so the idea is that we're going to do a prospective data collection. One aspect is to organize QC, so exactly what the SIG is supposed to be doing, which is define standard and have some ideas on how to compute things and so on and so on. But the other is also to say, well, how do we use the QC data that we will collect, and how we can do stuff to how does it affect group analysis for fMRI or diffusion, or also volumetry, which is what Derek has been published.
Yeah. Yeah.
We don't know what phantom we're going to use. We just, at the moment we are planning phase.
Yeah. I would, from the perspective of someone in industry, I think that it's great to work towards, instead of a specific phantom, properties of a phantom. In the end, to be widely used, you really want multiple manufacturers making these phantoms and selling them. And I mean, for your own purposes, you'll have a specific one, you'll have some custom phantom made, but to work towards specs on a phantom, that enable, different commercial entities to start making that phantom and selling it, and it would be a good endpoint.
Kind of the opposite what you are thinking about, Pradeep, but it's interesting, right? It's more like from what we can collect and what we can do, we can come up with some spec of what a good phantom could be. That what's missing from what we're collecting at the moment. Right? That's what Richard you're saying, right? Turning the other way around. Not asking you what a good phantom is, but more from what we can measure and say, well, what's missing. So the next generation of phantom should have these properties.
Yeah, I mean, for the application, you are the right people to tell us what needs to be in the phantom, right? And we're not close enough to each application. I'm not an expert in the type of things you folks are doing, but very interested in talking to understand, what it is that you need, and to see what the how widespread this would be. One of the challenges in making these phantoms and selling them is that these are small volume elements. It's actually a much higher tech device than it looks. It looks like a bunch of plastic and water. There's a lot more to it than that, and it's a small volume kind of thing. So by the time you go through all the development costs, and remember you have to have a whole quality system and procedures, and you go through audits and everything. It's not cheap making phantoms. It's always more expensive than people think it's going to be.
Is it $1 million or 10,000, 50,000?
It's not. It's not $1 million. It's a lot more than 10,000. It's generally a lot more than 50,000. But you have to look at the entire cost. It's just
right.
The point is, if it were if it were actually much cheaper than people sell phantoms for, by now, 30 plus years into MR, you would see all kinds of companies out there selling phantoms for much more cheaply, and you don't, and it's because it is a lot more complex than it looks. But getting back to the question of what's needed, I think it's always got to be working backwards, and I think the way all of us phantom manufacturers work is to collaborate with sites that are doing
right.
application specific. I think we do custom phantoms, and often that will turn into a commercial product.
Yeah.
If I could comment on, so at least in my case, I want to figure out what phantom should it be, which will be between those extremities, right? So there is study specific phantom, where you look for specific effects, right, to ensure that sequences work and everything. And it will be more expensive likely because customly designed. And on the other spectrum, like the stupidest, right, the ball, right, which doesn't give us maybe as much information as we want, and how to convince the centers which run different studies. So it wouldn't be per study specific phantom, not per protocol specific phantom, but to convince them that, oh, do QA with this phantom and you get the best bang for the bang for the buck, right?
Yeah.
So we need to figure out which phantom could fit this niche. It's definitely not in 10,000s range, right? It's something what technicians also, which wouldn't degrade over time as badly, because more complex phantom, more chances for it to fail, in some spot, right? So it should be something simple, which provides, and radiologists, they also don't like doing this. You said it, right? It's boring, right? QA.
Yeah.
So at best, what they would do, they would stick it in every Monday, right, into the magnet, run a bunch of sequences and put it on the shelf.
Oh, you're an optimist, are you?
No, that's well, that's what's happening in our center, right?
Yeah.
Right, that's the
So
plan,
it's-
Richard. Cyril, me, and I think David Kennedy at UMass, at least 3 of us are planning to do that.
Mhm.
So one of the questions I put it in the chat is, because it's a perspective study, we have the option of planning, like you said, work backwards. Sure, one of the goals could be, what would make a good QA, and
Yeah.
like you said, it's going to be complex given the diversity of the use cases. Maybe one of the things I want to target is fMRI, given rich history of the burn QA and whatnot, and T1 and T2, for example, right? So yeah, sorry, Eric, go ahead.
That's okay.
No, continue.
My point was that it cannot be really, it couldn't be really complicated thing. Otherwise, we will not convince those radiologists to use any phantom, or they would use something, but it will not be applicable to our study. So it should be ideally some phantom, which is not just a ball, right? But it's also not super complex, which maintenance is low, but maybe it doesn't provide all possible characteristics, right?
Yeah.
That's why to some degree, I like those Lego phantoms. They are relatively cheap to manufacture, right? They provide some geometry. They're not as good maybe as all the fancier ones, but they also probably degrade through time and nobody knows how.
Yeah.
But something in the middle, a ball with a piece of Lego, I don't know.
So Richard, how do you compare the fancy phantom you showed us in the talk to the Lego phantom? I'm guessing you're going to say that's better, but how is it better?
So I'm actually not that familiar with the Lego phantom, and I think I've seen a little bit on it. So this is a structure, a Lego structure within a phantom, and Legos are built to pretty high precision, right? So, yeah, I mean, so is this something that you build yourself, or somebody, there's some standard version of it?
A good question.
I believe there were originally Germans manufactured it, and then Canadians used it for their travel in phantom study, and now maybe they still use it across different sites to correct for geometric distortions.
Okay.
Vladimir Fonov was the one which taught me how, and I'm pasting a link in a chat which leads to preview of one of those images. It might take a little bit to download.
Yeah.
So, but
so a couple of things, when you're designing for accurate segmentation, there are certain feature shapes that are a lot better. You can detect edges very accurately. So certainly a Lego, phantom, you could do a pretty accurate measurement of that. But I think a lot of it comes down to just the practicality. If you want to build a phantom yourself and implement the analysis and do all that, you're probably going to end up, if you account for your costs properly, including the overhead of working at an institution, you're probably going to spend a lot more money than just buying a commercial solution. But also, our focus is really trying to bring a more widespread use of things. So if somebody just needs things for a few sites, or a particular study, more, more power to them. If they want to build, if they have a solution that works well, great. But there are a large number of sites out there who don't want to spend all their time doing that. They don't want to spend their time focused on the QC, and they really just need turnkey solutions for that.
Yeah.
Because there are a lot more interesting things, and we really need people who are specialized and interested in QC to develop the tools so that others don't have to spend so much time on it.
So I guess we'll have to communicate with you over time to see how your stuff differs from the Phantom, Lego Phantom. I guess cost is gonna matter. Like Cyril said, we're in the planning phase of the data collection. I'm personally interested in actually getting the best Phantom possible for this collection.
Yeah.
As long as it's in our financial means, because I think that will help us not only advance the field, probably help develop guidelines too. So
yeah. The other thing to consider is I don't know if what range of measurements you do with the Lego Phantom. Is it just distortion, or do you do a bunch of other things with it too?
To say the truth, I don't know. I know that it was used for geometric distortion.
Yeah, because one thing we often find is that a lot of our customers find great value in the fact that we've wrapped up as many measurements as we can in a single Phantom. Because before this product, those using this in radiation therapy had about 4 or 5 different Phantoms that they were trying to use to measure this or that. They have a distortion Phantom, and they have a uniformity Phantom, and and it was such a pain in the neck. They weren't really doing it. So they really appreciate having one Phantom that covers a wide range. And for widespread use, I think that becomes important. Again, for just a single study, you do whatever you want, whatever works.
You've mentioned that Phantoms might fail, right? What would be mean time to failure for your Phantoms, right? So how long-lasting they are?
Yeah. So our newer Phantoms are much better in this regard. The original AD&I Phantom had multiple fluids. It had spheres that were filled with a copper sulfate solution, and then just a more or less deionized water on the outside. That over time, over a few years, those spheres were prone to leakage. Now, for the most part, we weren't using those quantitatively. We were just using them to locate, so they could tolerate a great deal of degradation before the Phantom no longer worked. It depends on the application. We're a little newer into making the T1 and T2 arrays, where you're looking for sub percent precision. I don't know how long those are going to. That's one of the important questions, and it's one of the difficult things about getting into making those things. Characterizing that, and what's the reality going to be once you put it out in the field. So it's very dependent on what you're trying to do with it. With a single background fluid, we're not really relying on the exact value of that fluid. We just want contrast from it. And because we're supporting a diverse range of pulse sequences anyway, it's perfectly fine if that were to degrade a bit. Now, for longitudinal tracking, that's not true. And so if people are looking at signal to noise and seeing that slowly go down, we would certainly have to ask the question: is that the Phantom or is that the scanner? But we haven't seen that in our Phantoms have been out there now for several years. The first one, the big one, has been out for several years, and we haven't seen any signs of that happening. But in the old AD&I Phantoms, seeing now those are out there 18 or 19 years, seeing how many of those spheres have failed, it looks like it degrades over the course of the time constant of perhaps a few years. So some of the spheres, it's variable. Some spheres seem fine. Other of the fiducial markers, are fully they've lost all of their contrast. Clearly they leaked or the plastic wasn't high enough integrity. Something happened.
Richard, can I ask you, the ADNI Phantoms, are they make, you're not making those now anymore, are you?
We're phasing them out. We still, if somebody really wants one, we can make it. We encourage the more modern Phantoms. The ADNI is a very old Phantom and very limited in what it does. The more modern and the more modern Phantoms do so much more, and they do that function better. They measure distortion better as well.
So what would be the equivalent to the newer adenine phantom that would fit actually in a 32 channel. I think like there's a couple of groups that wanted to use one, and it just doesn't fit. The regular adenine phantom doesn't fit in a 32 channel Siemens coil.
So we have this, we call it the S162. It's a head-sized coil.
Oh,
It's
okay.
made,
I think you
head-sized
showed that.
phantom.
Oh,
Yeah.
okay.
Yeah, and that's the
you
one.
have you have those for sale?
Yes, yeah, we do.
How much they go for, like the MSRP?
They're like between 13 and 14,000 US dollars.
Okay, and then I mean, like, you give like certain discounts to different groups and stuff, like different universities, and
Usually it's volume, kind of volume. Also, prices in Europe may be different. We go through a distributor there, but volume, there's, you know, if people are buying several, then there are discounts. But yeah, it's usually more related to volume if people are buying several of them.
this would be, I think, maybe a multi-center study, like 3 centers, Pittsburgh, maybe Vanderbilt, and one other center. I mean, I think they were, I don't know if they, maybe they already reached out to you guys. I'll have to see, but okay, it's good.
And by the way, also for that you get 2 years of the analysis if the service, you know, the cloud-based analysis service with the Phantom purchase.
See. Okay.
So any other questions, guys? I have one more question that I want to ask before we wrap up, and I guess it's yeah, I guess so. The question I had, I guess I already asked in a way, you know, can we standardize between the two companies, or at least between industry and academia, so to say? You know, the big question that Richard nicely laid out, you know, is the scanner working well or not? You know, that requires, I think, a good working definition. I don't know if there is an accepted definition.
Well, I'd make a couple comments. So first of all, you know, Aaron's company and our company make very different kinds of phantoms. So I don't think there's, with the range of products that these two companies make, I don't think there's any sensible standardization. We're in, you know, rather different spaces. But you know, I think when working forward, it should always be about specs on a phantom, not trying to standardize and say you're going to make this phantom exactly this way, but lay out the specs, you know, for
no. So just to elaborate, what I mean is basically, like you said, we need to work backwards. I know the predominant goal for these phantoms is systems QA. Is the scanner working well or not, right? So what is that? Even when you use different phantoms from different companies, so to say, right? So if let's say if we're able to measure 20% drift, you know, is that when we say that it's not good anymore? When should we discard the data? That's the kind of question I'm trying to ask. You know?
Yeah, and that's extremely application specific, right? That question, you can tolerate all kinds of, like distortion, for instance. You know, you may be able to tolerate all kinds of geometric distortion in much of what you do, but a radiation therapist can't tolerate much at all.
I was referring to neuroscience, just fMRI, EPI, you know.
Yeah, I mean, even within, I mean, even say, you know, volumetric MRI studies versus fMRI, you know, everything is going to lead to different specs. So I think what you want is a tool or a set of tools that enable you to probe certain properties of the scanner, but what and you want the flexibility to set your own specs. You know, so you perform these measurements and then you say, for this application, this is what I need on it. And you you know, you don't make the phantom do a specific thing. You get a certain type of measurement and then you set your specs on the measurements from there for your application, and a different application will set different specs.
Yeah, that was kind of something I was touching on with our Neuro QA idea, really, is so that, you know, we can come up with some, you know, performance metrics we want to measure. We can come up with the way of measuring them involving a phantom and a protocol and an analysis pipeline and all that sort of stuff. And then the actual thresholds for where you say this is acceptable or not, that ultimately comes down to the application, you know, and that needs to be validated via, you know, some reasonable way of doing that. It might be that you have a, you know, series of MRI scanners, you will acquire these images over time, and you also say scan a volunteer, you scan, you know, a human phantom, and you either just wait for things to happen, or not, or you intentionally mess with the settings. You know, you can play with the shims, you can play with the RF, or you could do something, you know, you could open the door and have a load of spikes or something, and you can intentionally make the image quality worse and see if your test is diagnostic to that and where the threshold is. But this where, you know, as a phantom manufacturer, but neither I or Richard, without the MRI systems and without the tacit knowledge of, you know, actually hands-on what's going on, what is needed, we can't set that. All we can do is, if you say we want to measure these things, and we can think about how can we go about doing that and what's a suitable specification, and work with you on that to try and come up with a phantom and come up with the tests and that sort of thing, you know. But the actual thresholds do need to be developed. I think, you know, it's certainly collaborative, but they're also going to vary somewhat with different manufacturers, unfortunately. At some point, you're going to have to stratify. It might be at the protocol. It'd be great if it's just at the protocol level. If you can standardize everything, you know, once you've acquired the data, you do it in slightly different ways, but then you can standardize beyond that. I think that would be the best we can hope for. Anything, you know, beyond that, like, you know, the data looks a bit different, we've got to just do a little bit of data massaging to put it in the right format, that's fine. But if you end up with different QC parameters for Siemens versus GE, that becomes a problem.
Can you elaborate on that, Arun?
Well, I mean, if you have a metric where on a Siemens the threshold is here and on the GE the threshold is here for acceptable data, it's not a very good metric, is it? Ideally you want something that is standardized between them all. You know, so it might be like gradient linearity is probably a good one. You know, that's probably something which is it's more in sense of you're measuring a physical parameter or for a physical quantity that's related to the MRI scanner, rather than something which is a bit wishy-washy and it's a combination of multiple factors. That's the thing. If you're measuring something which is a combination of multiple factors, then you're not going to necessarily be able to standardize that across different you know, systems. Even the same type of system might, you know, in different places that might not be very good because they've got different, you know, factors that affect it. I think, you know, I mean, I raise this question is that can you compare your FBurn parameters between systems? You know, is that something which is done? Are people going, hey, we're getting this score, what are you guys getting? Or do you just look at what's the relative change?
Good question. Cyril, Yarek, want to answer?
I don't have an easy answer.
So I mean,
Sorry, the answer we have is the metrics that works best are always relative metrics, right? Ratios is the difference between two stuff. Absolute metrics, they always change, right? If you say, as you were saying earlier, what's my actual value of diffusion or perfusion? Quantitative stuff, that changes between systems. And it's very hard. We thought it would not change, right? If again, if we say like a T1 time should be the same because, you know, the time to relax is the same. Yet between systems is not as consistent as we hoped it would be. Whereas if you do a ratio like SNR, when this is relatively consistent no matter the system, because maybe one system has overall higher values there, but the ratio between two, it works. So some metrics clearly are better than others. I agree with that. Some metrics are good for your system and cannot work only on your system, and you will not be able to compare with the neighbor. That's very
Should we
unfortunate.
maybe aim for quantitative MRI based QA then, right? So because T1 weighted is indeed many factors affected, and that's why quantitative MRI is supposed to normalize it, right? So you have specific
even
things.
so, it's not as good as we thought it was, because for instance, the temperature would affect your T1 time.
It's not just that, but you have an additional propagation of errors because of you fitting to a model. But, and I think, you know, the fundamental thing with you know, just relaxometry, T1 and T2 are not, I mean, you know, NIST is, they've got them related to SI units, but actually, you know, these are molecular tumbling times but there are other mechanisms, you know, paramagnetic, you know, relaxation, all those things. You've got a phenomenological number. which is dependent on a number of things. And then if say you're measuring T1, depending on what kind of pulses you're using, you're going to have different amounts of MT, which is going to change the measured T1. T2, I mean, NIST, they quote it based on the refocusing time of the pulses. If you measure it differently, you'll get a different number. Some of them are better, like diffusion is in principle better, but it's apparent diffusion coefficient. It's not the actual diffusion coefficient. It's going to depend on other things, chemical species, all sorts of things like that. So I disagree that for QA, you should be using quantitative, like QMR parameters that you're trying to measure in your patients. I think instead, you should be trying to benchmark the MRI system and the hardware of whether are the coil elements failing? That's a really important one. Are your gradients linear? Are you having stability and linearity in your RF transmitter? Are there issues in the receiver chain? Those kinds of things. Are there other things that are problems? Is the table shaking, because of the high diffusion? Is the gradient amplifier failing when you run the really high duty cycle sequence that it should be able to run normally? These are indicative of there's a problem that is slowly developing with the MRI system. And I think that's, if you can detect that early enough, and not, because ultimately, if you've got images which at first glance look okay, or maybe there's a bit of a difference in homogeneity, or the SNR is a little bit poorer in this region, that will get through the test. People will say this patient data or this subject data is fine. It'll end up in your cohort, and then at some point somebody will analyze the data and realize the numbers are completely wrong, and then you discard the data. That's the important thing if you're doing neuroimaging studies, really, is you don't want to discard your data. That's the purpose of the QA. For clinical practice, is different, because it's at what point do you say that this is not deemed good enough for diagnostic purposes? And that bar is pretty low, because radiologists are very good at seeing all sorts of garbage in the images, patient motion, all sorts of things. So actually probably the physiological noise is far higher than what you would have from the system, unless there's a real problem. But when you start doing the advanced techniques, that becomes a problem. And when you do things where you need more precision, like in the radiotherapy planning, those kinds of things, that's where this is necessary. But I think that's where you need to approach this from, in terms of defining a specification, is going to be able to make this technique work and get the data that I need for my studies. We need to have a system that is working in such and such a way in terms of the performance of the hardware. Ultimately, it's that's at the level that you can detect in QA. You also need to do acceptance testing to make sure that a site is able to actually generate the data that's of a high enough quality. But that's not just, is the MRI system okay? It's, are they following the procedures okay? Are the staff trained correctly? And I think there it is useful to have a phantom or a human phantom, but actually where you're measuring what you're trying to, what is needed in the study. So that, like, for example, you want to measure perfusion, having a perfusion phantom or a volunteer where you tell them not to drink coffee, and you don't, get a good night's sleep beforehand, so that you're going to be able to get some good data, is reasonable. But for actually working out during a study, are we getting consistent, is there an issue with the MRI system? There's probably a better way of doing that than scanning a perfusion phantom every week, because the technologist is going to see that as another annoying thing to have to do, especially if the phantoms are heavy or hard to use, or things might fail, or it's too much, really. I think that's my feeling on it, really.
I mean, we do plan to acquire a phantom once a week, but at random times, not specific time of the day. Like, at some point during the week, we need to acquire half an hour phantom data. That's the plan.
I would suggest scan the same time every week and do a random time.
But that's not what Yannick suggested, because model variations over time, over like a year or two, it turns out that from the discussions we had before, right, Pradeep, was like some people show that if you do your scanning Monday morning on the fresh scan, your parameters values are not the same as on Friday evening, except that you scan patients and subjects for your studies all week long. So you want to have a range of variations. You don't want the always freshly shimmed and freshly ready scanner from Monday AM.
Yeah, I think Aaron is right. We need both probably, to avoid any issues having to do with the interpolation correctly. Standardizing the time is good, but also randomizing is helpful to capture more variation.
Probably need
but that,
two, I think is what you said. It depends on what you want to do,
right?
On your goal. If you test the scanner for the scanner, but always the same time, Monday AM, for instance, at first thing, is probably good. If you want to control for variation in the scanner for your studies, or your group studies, then variation over the week is better.
Yeah. So
because it affects what you do.
Randomization helps remove any batch effects. For example, if there is some kind of hardware maintenance happening on Sunday evening before they resume operations, we're not really catching any issues that are happening downstream in the week. Maybe Friday evening is a better time to before any maintenance. Proud, does that happen? What kind of maintenance happens weekly here? Or
Weekly, I don't know, daily, I mean, daily QA. But I did have a question for Aaron. So, I mean, I understand what you're saying with the main focus of the QA should be, well, is the scanner running? I mean, rather than like looking at the sequence, like, oh, because the empty pulse may be different on a Siemens versus on the GE, so you can't quantify for that. But assuming you're past that point and, you have two scanners or more than two scanners, more than one scanner, so 2 or like 10 and each one of the scanners passes the QA. I mean, like all the QA result in measures that are within the range, expected range. And so do you see a purpose for the quantitative MRI where you calibrate the scanners? So for a multi-center study, you would be actually looking at how do the results on one scanner compared to the results on this other scanner, like from a different manufacturer, like even though the sequences may have like different, as you mentioned, like different, MT transfer pulses or like whatever, different implementations. So where do you see because I think a phantom, I mean, will still be very useful. I mean, in that scenario.
I mean, you could characterize it, certainly if you've got a system phantom of some description, you can characterize what the differences might be. The thing is that what does that mean for your patients? Like, if basically, yes, you can put a sphere in and with lots of samples, like say the Calibre MRI system phantom for relaxometry or something, where it's you've got all the NIST values and everything, it's characterized, it's, and it tells you the difference between the different MR systems under these ideal conditions is this. But what does that mean for your patient when as soon as they go in there, they affect the shim, they affect the RF homogeneity, all these things. So there's more to it. The other thing as well, so it's a real shame that he didn't publish this, but somebody from the National Hospital at Queen Square in London, so he presented this a few years ago. It was a QMR thing at MPL, where they said, right, let's try and see, can we genuinely get quantitative T2 values that match with what we have from our NIST phantom, in terms of what's in the manual saying the value should be this. And so what they did is they had like a toy sequence, which was, your multi-echo CPMG type sequence. They then fitted it to a model that took into account the slice effects. So, the imperfect refocusing. They also looked into how do they optimize the actual fitting of the parameters. And they managed to get, results that were nice and close within probably expected sorts of, margins of error to what the values from NIST were. But then they were like, okay, we don't really know how we can then apply this to an actual clinical sequence, because we're not going to do a single slice multi-echo, spin echo, CPMG sequence for measuring in patients. This is not feasible. This is not possible. And then suddenly you've got much more complex physics. to actually take into, and yeah, you could just say, well, there's some fudge factor, but the NIST values should be this, it's this, so we just adjust it to do this way. But is that making it worse? And every stage of processing, you're adding more uncertainty and you're propagating your errors and your noise through. So that's another thing to sort of worry about. I think that I've seen a couple of things where people have done that, like for MR fingerprinting and things like that. And I think you have to be very cautious with that kind of approach. Unless you have a reference in the image with the patient while you're scanning it, I wouldn't trust. I don't think you can do it. I mean, we literally have our SPI project at the moment. In the grant, we said we were going to do this with our ASL Phantom, and said we will calibrate the ASL using the perfusion Phantom, and then do some image correction on the patients. And actually practically, I don't think we can do it. I don't think it would really work at all. And like, how do you go about doing that is to be decided. I think it would take a lot of work, and you have to address some of the smaller issues to begin with. And then if you look at like the Kiba profiles, which are basically providing recommendations for doing quantitative MRI in clinical trials, generally they seem to all follow a similar approach, which is we recommend a quantitative calibrated Phantom with NIST certified or some of the National Measurement Laboratory certified values to demonstrate that you can get sufficient data quality at site qualification. And then there'll be some sort of recommendation for ongoing QA through a study. But they're only recommendations as well, because they can't mandate that people buy a Phantom. I mean, people probably will, because it's the way of addressing the problem and demonstrating it. But these are, I think Kiba is probably they are pushing in a certain direction. That's probably where everything's going to go. And I think they are that's the right. I think it's the right sort of approach practically. I mean, I'm leading one of the task force for the ASL Kiba profile on QA and Phantoms.
I mean, I think for just one patient, or for the patients who get scanned on that particular scanner, I mean, like, it doesn't make a difference. But like for the big multi-center studies, I mean, like where you would be able to actually combine data from different scanners, different manufacturers, possibly different field strengths, harmonizing data. I think, I mean, like that would be, a Phantom, and I think Phantom would play, an important role there, not just for QA on that particular scanner, but also calibrating.
If the differences are due to sort of static offsets between the MRI systems and are not due to the interplay between the patient and the MRI scanner, then yes, you probably could. But it would need to be quite well characterized. And I think you'd need to have a really good handle on the uncertainties. Otherwise, I mean, again, metrologist hat on, we should all be worrying about the uncertainties in quantitative MR and reporting these and that kind of thing. And that's not just, oh, I did an ROI and this is the SD. It's the actual uncertainties in the measurement. And that means if you've got a Phantom, you're starting with the uncertainties in the values of those and thinking about how that all propagates through. And I think also that means you need to really know a lot more about the pulse sequence, which is quite tricky with some of the vendor sequences as well, to know exactly what the physics are.
Right, but if you have your own pulse sequence.
If you have your own, sure, but then you might not be able to use that very easily in clinical trials, depending on exactly what or on patients,
True. Well, thank you so much, both to you and to Richard and to Pradeep for inviting me. I gotta go, but thank you. I mean, they both have been very interesting talks, and thank you for answering my questions. I mean, discussion was great. Thank you.
Oh, thank you.
Yeah, thank you. It's been great. It's
Really
great discussion.
Yeah, thank you. I think it's been like 2 hours and 20 minutes, so I should probably yeah, anything else you guys want to say? I think that I would like to invite you guys to kind of draft all these ideas and discussion into some sort of Phantom spec, or like you talked about acceptability testing. I think this could be very useful, even for we can probably cover everything, at least for common use cases with well established lines of research like fMRI and maybe the lessons you learned from the ASL effort you were involved in, translate that to fMRI, you know. Yeah, so I would that be interesting to you guys to I'm certainly interested in trying to use one of your phantoms to see how we can improve value of our data collection effort with just planning to do the best, and how they can help us develop standards for our analyses. So maybe there more needs to be discussed, you know.
I'm very happy to.
Yep, likewise.
Awesome. Yeah, thanks, Richard. Yeah.
Yep.
Yeah, maybe Eric, is that it? Should we call it a day? I guess it's probably too late already.
I think that was long enough, man.
Thank you so much for taking the time.
Thank
and
you.
Yeah, looking forward to continuing this discussion. And if there is any code / data that you would like to share, please let us know. We can put it on the website and make sure everybody in this space knows about it. You know, I'm especially curious about the digital reference object that you mentioned. I think that's a very nice way. We should do more of that for our, I don't know, even for the quick question. Have you guys looked into these Burn QA QC scripts, making sure they're doing the right thing? You know, I'm guessing that they're really old software that could use some help from us, you know?
So we, you know, what we have on our website is basically, we did quite a lot of looking into it, and like there's differences between what's in the paper and what's in the, you know, there's a PDF floating around on the internet which kind of describes it. There's a difference in what's actually calculated. So we decided to follow the publication rather than that thing. But yeah, we've implemented tests and all sorts of stuff like that and validated it. So there's, you know, but one
Do
thing
you
we
consider
were
that a trade secret?
No. I mean, I think, yeah, it's something we can discuss probably, you know, later down the line about how we go about doing this, you know, I mean, but yeah, it's the next step for us was to make a digital phantom, actually. And at the time, this was a couple of years ago. And then at the time, we didn't have anything as a framework for a digital phantom. Now we do. So we could, in principle, do that. And, you know, the thing is that what you really want to do is we start from the image, the quality metrics, and synthesize those and you know, put that into our digital phantom. And then at the end of the processing, we should be able to get the same number or a similar number, you know, based on the kind of what you'd expect from the processing pipeline. But, you know, that's something which if there is interest in doing that and using our I mean, I'm already thinking ASL DRO makes it sound too narrow. It probably should have been neuroimaging DRO or just MRI DRO.
Yeah, there is a lot of space for innovation there, you know. One thing I want to do is have a common library that everybody uses, or at least common tests that different implementation can be evaluated against, so they're actually computing the same SNR, you know. So my sense is that they talk about, like, you've probably seen this discussion at the SIG Google group, you know. There is no standards for SNR calculation, you know. So, and that, yeah, like for example, that dynamic phantoms paper that I shared, you know, they make the point that the SNR calculations are not even comparable across scanners for reasons they explain that I don't fully understand yet, you know. So I'm trying to reduce the scope for our uncertainty by using same implementations / our comparable implementations, you know. So the any software modifications you probably made in GSP Cloud or Phantom Lab Cloud, we would be interested to know why and how we can actually just propagate it back to the community, so we learn from your experience, right?
Sure, no, absolutely. Yeah, I think if I remember, a lot of the issues really are to do with the fact that the data that comes in looks very different depending on MRI system. I mean, you probably have the same problem.
Right.
Actually. It's just, you know, it's not the actual image processing. Once you get it into the right format, the image processing should in principle kind of work, but it's, and you know, but it's getting it to that point is the most challenging thing. I mean, the ASL was an absolute nightmare because
Actually,
it was
yeah,
so fragmented.
Right, that reminds me of our previous discussion where we thought there needs to be a BIDS-like standard for phantoms, you know. Is there one? There isn't, right? You know,
I mean,
so
I
one
don't
of
know
the-
if I don't know if there necessarily needs to be a bid standard for that. I mean, I went around this with for DROs actually. Should we have some sort of standard for DRO data? Because I mean, obviously the ASL DRO outputs in BIDS format, and the question is what format? How do you want DRO data to be? Do you want to know it's DRO data, or do you want it to masquerade as real data? You know, because depending on the use case, so there's a few different ways. Also, you want to know stuff to do with the provenance of it. I think that probably with phantom data, the stuff to do with the provenance and the traceability of the phantom is important, but I think that's very hard to link to the image data unless you either have something in the image, you know, and there are some we don't do it, but there are certainly some manufacturers I've seen, they put QR codes or, you know, a MR visible ID. That's one way of doing it, although it won't be in every image, every slice. And, you know, DICOM files are one image per slice in you know, the traditional format. Or, you know, some other way of having traceability, which means you need, you know, some way of linking it to the MRI console so it gets the information in there. So I think that's not there. Having some sort of standard or, you know, way of presenting the data is useful. But it's, you know, to me, I think the traceability is also important, but, you know, we haven't sorted that. In our Casper Phantom, we actually have some additional data that is generated by the Phantom, which is the flow rates and things like that. And that's, you know, taken, and that kind of provides the information about the Phantom, the serial number and things like that. But that's quite a special sort of use case. And that's because it, you know, that's the dynamic data that confirms the Phantom is operating as you want it to.
Yeah, I mean, like one of the challenges we identified in the last Phantom discussion was the challenges in linking a correct Phantom to the right human subject data, you know, because of the difficulties in sharing dates with the data in DICOM and whatnot, you know. So if they were acquired separately and if you want to combine them later, there is a fundamental problem because there is no obvious direct link without the dates and whatnot, right?
Mm-hmm.
So one of the solutions I was thinking, maybe I don't know if it's a solution, but is to package them in a way where you don't have to share the dates, but there is correspondence and link between the two, you know. Yeah, that's where I'm coming from, you know. If it's all where if it is all just phantoms alone, then it's not a big deal. But when you're linking to humans, some sort of linked standardization would help too. Am I communicating that correctly, Eric?
I guess, sorry, I phased out.
Okay, yeah, no worries. I think, yeah, anyway. So yeah, so thanks. I think we should probably call it a day. I'm also hungry and
but I wanted to add, thank you very much for doing this business. You know, people just don't appreciate all the phantoms, especially in research, right? For clinical, you know, thing, yeah, sure, you know, they don't want to be liable and, you know, they gain good buck for it. But in research, it feels like all this phantoms QA is just really undepreciated. So thank you very much for doing that. And Pradeep, we should have a page somewhere, right? If unless we already have with companies like this, right? And maybe key points and key phantoms, which are the best kind of feed for research projects or what types of research projects, right? Let's say longitudinal studies or big center studies, right? So which phantoms we would recommend, and also point to any of your kind initiatives to share stuff like code and protocols, right? So we could promote you because I think you're doing what is needed to be done, right? And others consider boring and not really useful, maybe.
Right. Yeah, so I think I did share this with the group. I created a page on our SIG website where we list a few commercial solutions. Perhaps I can take Richard and Aaron's help to improve them. I think they shared a lot of useful information. I much admire their commitment to improve the state of the art besides just selling their products and services. So this is just a markdown file, Aaron and Richard, if you want to write more in terms of why they should use your phantoms or the different efforts you are involved in helping the science, please go ahead, send a PR or let me know. I can add some stuff,
Thank you.
Yeah.
Thanks.
So go ahead, Richard.
Oh, no, I just said thank you. Yeah.
Yeah, well, I'll certainly let people know. I have some understanding of what's going on. I'll probably watch the video again to better understand it. So yeah, anyway, thank you so much. Much appreciate your time and effort. Let's continue the discussion in the Google group.
Thank you. Thank you, everyone. Yeah.